coming, everybody. Today's talk is by Shameer. He's one of my PhD students in our research group. And so he joined us from Cochin University. And well, I first heard about him by email in 2010 from Cochin University. And then he spent about 9 months fighting with university bureaucracy and Immigration New Zealand trying to get a visa and all the associated dramas. And he persisted. And he turned up 9 months later, I think. So he's going to tell us what he's been doing in the last 4 years, I guess?
Yeah.
3.5?
3.5.
3.5 years with his PhD, so.
3 years, 8 months, I guess. Yeah. Yeah. So welcome everyone. This is supposed to be my PhD exit seminar, but I'm quite not sure like if my PhD has exceeded because my thesis is currently under examination, so it's kind of So my talk is on my thesis is on high-performance computational simulations of gastrointestinal electrical activity. Leo and Mark were my supervisors. So I'm associated with the gastrointestinal research group. Okay, so gastrointestinal electrical activity. So, this is the motility or the contractions that happens inside the stomach, which is useful for mixing up the food and breaking down the food down to food particles and mixing with proper enzymes for digestion when entering into the intestine. So as you can see that the muscle contractions start around, so this is inside the stomach, human stomach, so this starts as a form of, it forms a ring and starts from contracting at the top and it moves along the stomach towards downwards. So what causes this muscle contraction? So this is something called interstitial cells of Cajal, which are called the pacemaker cells, which has the capability of generating electrical activity. So this electrical activity is passed on to it's conducted to surrounding smooth muscle cells, which actually contracts the muscles. So that's how everything works, like based on the importance of electrical activity. So as you can see on the top right-hand corner, so this is the intracellular ICC and smooth muscle cell recording. So it's like simultaneous recording. So from this, it's quite obvious that the ICC generates first and smooth muscle cells are affected later. So how are the ICCs and smooth muscle cells, it lives inside the stomach or gastrointestinal tract? Actually, it's connected through gap junctions. So this is from a paper from 1997 from Ishikawa et al. So as you can see that interstitial cells of Cajal is here, that is marked in there. Then this is the gap junction between the smooth muscle cell and ICC. It can be possible that there will be a field coupling, so whereby electrical field can initiate the activity onto smooth muscle cell or other processes as well. But this is kind of like agreed sort of thing. Now the ICCs, so like if you refine further into ICCs, ICCs are kind of arranged as a network along the stomach wall. So on the left hand side you can see the healthy network, and right hand side you can see the depleted network. So it's quite you can like visually see that it's quite depleted. The ICCs, there's one particular feature of ICC is that it has an intrinsic frequency which actually varies over spatially. So for example, say this frequency is higher at this corner, and over here this frequency will be lower. So the frequency at which it generates electrical activity. But when we consider as an intact network or in a healthy network, all these ICCs are entrained to a single higher intrinsic frequency. For example, in this case, in my example, that would be the frequency of this corner ICC. So that process is called entrainment. So it has been already proved that several motility disorders are associated with ICC loss and network degradation. So that's the basic understanding of how ICCs work and its importance in the gastric electrical activity. So in human stomach, like as a brief review, slow wave activity, or otherwise it's the electrical activity inside the stomach, which happens every 3 cycles in every 1 minute. So it starts at around the corpus region, like the upper part of the stomach, and it forms kind of a ring, and it moves towards the downwards around the stomach. So there are at times like 3 to 4 rings forming at times. Abnormal electrical activity is associated with gastric motility disorders such as gastroparesis and functional dyspepsia. Gastroparesis, for those who don't know what gastroparesis is the severe cases of these might include nausea, vomiting, bloating, severe pain, and usually patients have frequent hospital care and poor quality of life. Gastroparesis is present around 25 to 55% of patients with diabetes. So considering the current availability of fast foods that we have, it's kind of we have to be delving into it deep. So how to treat gastric motility disorders? So as people have been working with in cardiac field, as many of you here have been working with cardiac field, right? So application of external electrical impulses has shown to modulate or improve the conditions. So there are several ways by which gastric electrical stimulation has been applied in gastric field. So it has shown to be have therapeutic curing effect. Most importantly, gastric pacing, whereby you actually apply high energy, low frequency signals like LF. So the pulses would be around 300 millisecond width and of high energy, and that will be applied onto the stomach, which can actually modulate the underlying slow wave activity. There are other ways of adding the electrical activity, external electrical impulses as well, like for example high frequency, low amplitude signals, so which are kind of assumed to modulate the neuronal activity of the ICCs or the underlying physiology. So there is a couple of more other ways, like for example dual GES, where dual gastric electrical stimulation, where you have both high energy and low energy pulses acting together. And there are further one more, that is the applying the electrical activity to further increase the contractile of the stomach. So you have the slow wave coming in, you apply an electrical impulse from outside, such that the net effect will have an improved contractile activity. So all these are kind of an assumptions, and well, it's not an assumption, well, it's been shown to be working, but nobody knows like how it's actually working and what is the main understanding or underlying biophysics of this thing. So objectives of my thesis were kind of to develop a mathematical model to simulate gastric pacing and understand how the pacing protocols have the effect onto the underlying slow wave activity. So I divide the thesis topic into around 3 subtopics. So first we will delve into cell level biophysics. Then I'll move to tissue level, where I'll be applying the cell level description that I had developed onto finding the network structure function analysis for postnatal development of ICC networks. And then I move on to whole 3D network, where 3D stomach geometry, where I'll be modeling gastric pacing onto it and understanding the interaction of gastric pacing onto the normal electrical activity. So in this case, I'm just considering the high amplitude and low frequency cases, and not the normal other cases of gastric stimulation. So ICC model. So if the first one is the cellular level biophysics, right? So ICC models. So there have been quite a few ICC models exist, but there are only two of them that has shown to be that has been developed for modeling entrainment process. So as you know, like as I said before, the entrainment process is necessary for coordinated slow wave activity. There were different issues and limitations for these models. Some of them were the intrinsic pacing frequency of the ICCs being a major contributor to defining the conduction velocity. another one was none of these models were able to incorporate the effects of external stimuli. And then these models, when incorporated on a tissue level structure, it wasn't able to model the electrical anisotropies that's observed in the stomach. The precise mechanism of how this ICC entrainment happens is currently still a subject of investigation, but it has been widely accepted that the voltage-dependent IP3-mediated calcium release is one of the pacemaking mechanism, or which helps one cell to communicate with the neighboring cell. So what we propose is a voltage-dependent calcium dynamics using a finite state machine approach, whereby we modulate the calcium dynamics based on two states, that is active and passive. So we divide the entire cell process into two states, and then we kind of move between these and move between the two states based on the calcium dynamics of the cell. So this is the original ICC activity. So this is a single cell. Of course, we don't need to model entrainment when you have single cell. So this is the depolarization of ICC, and as it is it has an intrinsic pacemaking capability. In this case, it's like 3 cycles per minute, so it activates by itself without any external application of stimulus. So the dotted line corresponds to the calcium activity. So what we did was we divided these two as a passive and active state. So active corresponds to when you have this depolarization, and passive state when it's on the quiet state, or quiescent state. So it's quite clear from the study, from the figure, that during the passive state, the calcium dynamics at the quiescent state. So what we did was this is the electrical equivalent of the ICC cell model, or any cell model. For the time being, let's consider that. Or let's consider it be ICC Koreas and Bush model. So we only include the entire dynamics when we have a certain finite state machine condition satisfied. But when these conditions are not satisfied, we will be only considering the membrane capacitance that's acting in between the cells. So how does this finite state machine work? So that's the next step. So when this thing is included, then that will be, when this entire ion dynamics is included, that will be the active state, and when it is excluded, that will be the passive state. So that's how we have modeled the system. So this is the finite state machine state transition diagram, which controls the inclusion and exclusion of the ionic dynamics. So we assume there are 3 states. One is the initial state, when the cell is started to, maybe the beginning of the cell or something like that. This is the active state. This is a passive state. So we defined certain variables. Don't ask me why I chose DC, VP, and AT, but DC, I defined that to be a variable which actually traces the calcium dynamics that's happening inside the cell. VP is actually checks if the diffused voltage or diffused transmembrane potential is greater than a set threshold value that we define. Then AT is the intrinsic frequency. So you have the passive duration and also the active duration. So AT is the intrinsic frequency set parameter. Now ST defines the initial activation, which only happens, which is only relevant when the state is in the initial state. So I will define, I will kind of explain how things work. So in the active state, if it is in the active state, that means you have the transmembrane thing. It will continuously check for the calcium dynamics. So that's this one. If the calcium dynamics has not reached the quiescent state, it will be still doing the entire calculation for the ionic. Will be taking into consideration all the ion channel dynamics that's happening there. When the calcium dynamics switch back on, it switches to the passive state. So that's this one. And it remains in the passive state until you have the so it has the intrinsic frequency until the intrinsic frequency time has reached, or the diffused membrane potential has been below a threshold potential. So that will remain until that point. If not, it will be switching back to active state. So that's how this state transition diagram works. So this is actually the comparison between, so we took the original Curias and Buis model, ICC model. It's a well-established model. I should have told that before. It's okay. And this is the finite state machine model. So we did a comparison between these two. So this is only the ionic current values for both of the models. So there is the quite variations are there. Over here, as you can see, when you apply a stimulus over an active state of the cell model, and over here as well, you could see the same response. But during the passive state, you could see the activity to be shifted, and here it doesn't have any effect. So, and we also did a long-term stability analysis where we tell the cell model to run for quite a long while, and we apply stimulus at a set frequency such that the frequency is changed, and then we remove the stimulus, and then it returns back to this original state. Now there is, apart from that, its other capabilities, one other improvement was the simulation time. So as you can see that since we are not considering the entire ionic dynamics during the passive state, we actually see the finite state machine model to be very efficient, and it is to be dependent on to the number of active phases does it have, or passive phases does it have. So this is for 120 seconds of simulation time. So it's a time in seconds. So this is the finite state machine model when coupled, and so we have a 1D array of cell models, the cells, ICC cells, and this is when everything is coupled together, like all the cells are connected together. So, but I do have an intrinsic frequency gradient from 3 cycles per minute to 1 cycles per minute. So as you can see that when it is connected, everything is connected together. You can see that everything is moved on to a single frequency and trained to a single frequency. However, when we have like when we decouple the segments, like we have decoupling two different regions, which is actually shown in experimental studies, that if you cut the stomach transect the stomach into 2 different sections, then you will have 2 different frequencies of activity that's happening. So it kind of replicates that. So this entire activity will be entrained to the ICC frequency at this point. So that's this one, and over here as well, the entire segment of the ICC network will be entrained to this frequency at this corner. So that's the one. Then once the activity is entrained, like everything is entrained, if you apply what do you call the pacing or external injection of current at a set frequency, it will be taking over the ICC normal activity, as you can see by the arrows. So the length of the arrows represent the backward propagation. So now the next stage was to validate it with experimental observation. So what we did was we did a study in 2010, which was published. That was the gastric pacing mapping of the high resolution mapping of the gastric pacing activity. So we placed the electrodes like this, and the 2 needles of the pacing electrodes over here, and when we start to apply the electrical impulses, it generates an activity which gradually takes over the normal activity that comes from top to bottom. So after some time, it's kind of like takes over entire region. So this is the simulated activity. This is the experiment observation. So as you can see that there is considerable, it's in reasonable agreement with the experimental observation. So each of the color band in the activity is of 2-second interval. So it's the activation map. So we simulated successfully external stimuli, entrainment by higher frequency pacing stimulus. We captured the effect of anisotropic conduction over here. So it's like, as you can see, for one direction it's quite fast compared to the other direction. The model was stable, most importantly, because in normal cases the model parameters tend to deviate from the physiological parameters and cause issues, stability issues. But over here the model is stable because we do take into consideration the model variations and try to bring it back to the normal case. And also, most importantly, we obtain a major gains in computational efficiency. Now this is the tissue level, so we move on from the cell level to tissue level. So we have the cell ready. Now we could just kind of like utilize that for analyzing some tissue level aspects that is relevant to which is currently not research questions which are currently not answered. So this is the this is the postnatal ICC networks at different ages. So P0 defines like the postnatal age 0, 2 days, 5 days, 8 days, 12 days, 16 days, and 24 days. We had so these are the ICC networks I shown you when I started my presentation. So the red ones, I think it's not quite clear, but the red ones correspond to the ICCs and the black ones correspond to non-ICCs. The bottom 2 rows corresponds to the zoomed in version of the small squares in here. So we had like about 10 images at an average of each of these different ages, and we wanted to understand like how the ICC network changes during this postnatal, because soon after the birth the gastrointestinal tract first depends on the mother's milk, and which kind of after a certain stage it has to move to the solid to ingest, right? So during this process the ICC network undergo a huge amount of change. So we wanted to understand what is actually happening in the structure, and also understand the function, that is its effect on the pacemaking function of the ICCs.
What animal?
It's mice.
Mice.
Okay. Yeah, it's mice. So we did a structural analysis on one of my colleagues, Jerry Gao. He has finished his studies from here. So he performed a structural analysis, and he found that so this is his conclusion. So he found that the ICC remained immature state until P5, that's the age 5. From P5, that is from over here, and overgrowth of ICC is observed until P12, age P12. Then by P12, the selected ICCs are retained, and some are selected ICCs are retained and strengthened while others are discarded. So this is assumed to be a kind of like a pruning mechanism that's happening in as similar to the central nervous system axons. So they also form like a kind of around the similar way. They kind of first try to make the processes larger, and then tries to do a selective removal of some of the processes to get the efficient structure. So a similar kind of approach is hypothesized over here as well. Now by age 24, the new processes that are formed gets adjusted to the adult morphology. So as I told before, the central nervous system maintains the development of a functional neuronal circuitry through a robust mechanism of initial excess overgrowth, followed by selective pruning of axon branches. Is there a similar pruning mechanism in ICC as well? So that was the question. So we had to perform slow wave activity, pacing activity, normal ICC pacemaking activity. For that, we actually developed a modeling framework. So we obtained the images, we convert that to two-dimensional bitmap. We sampled the images. The images were around 0.5 millimeter to 0.5 millimeter in size, very small, and it is sampled to 1024 pixels by 1024 pixels at a resolution of 519 micrometers, so which is very, very small. So this is the geometrical representation. So this is the zoomed in version of this small block, and this is the same this block. So each pixel is over here. We obtain the triangular mesh by connecting these pixels together by form of a tetrahedral triangular mesh. So all this model development is done in a chased computing package. So it's being developed at the Oxford University. It's actually set up a libraries which we can use to kind of adapt to our simulation. So it's built upon MPI based libraries as well. So it enables parallel computing. So all the models were built upon this chased library. We set the red patches or red pixels to ICCs and black ones as non-ICCs, and we simulate the electrical activity. So this is the calcium activity. So we allow the ICCs to activate at this top right corner, and we see the propagation. So this is like a sample simulation propagation. So it's only the intracellular calcium. So we obtain we quantify the transmembrane potential that's observed from the propagation, and also the intracellular calcium concentration for different networks based on 4 parameters actually. One is the activation time, activation rate, and half peak calcium concentration time ratio. Well, this one was insignificant, but these two provided a very good understanding of what's happening within these networks. So activation rate, it defines the network area by the time taken for the average VM to reach a threshold of minus 30 millivolts. So as you can see from here, at the age of P5, I should have the, yeah. So at the age of P5, as I said, like from the P5 to P12, an excessive overgrowth is observed during the structural analysis. So from P5, as you can see, P5, the activation rate is quite low, and also the time to peak is quite high. That means it takes quite a long while to reach this activation rate. So from P5, the activation rate or the total efficiency is increased until P8, P12, and P16 as well. So until P12, it actually reaches a more stabilized state. Like from there, it's not steep. As you can see over here as well, time to peak as well is reduced. That means it reaches the required activation time in very low time. So that's kind of like suggests that the efficiency, that overall the network efficiency has increased, and it's increased over when it reached P12. Now over here, during the P12 and P16, as we had observed that the selective process, some of the processes are retained, while others the redundant ones are discarded. So does that have an effect on the total computational efficiency? We didn't find any. So it is actually doing the same thing. While maintaining the efficiency, it is actually removing the excess ICC processes that was in there. So it kind of suggests that there is indeed a pruning like mechanism happens inside for the ICC networks. So that's kind of summarizes what's drawn here. Initially an abundance of ICC processes are initially overgrown to achieve higher pacemaker activity transmission efficiency. The less efficient redundant processes are discarded without impacting the overall efficiency of the network. The retained processes are strengthened to further elevate the pacemaker activity output volume. But then over here, like we see, like the activation rate is reduced, and again for the structural analysis, we found that new processes were generated, and we believe that the development of new processes is in response to the myocentric neurons which adapt morphologically and electrophysiologically throughout postnatal life until adulthood. So we believe it's that one. So until the age P5, it remains immature. From P5, an overgrowth of the ICC networks happen. Then P12, it's indeed a pruning mechanism happens whereby the efficiency is maintained, and then finally it adjusts to the adult morphology. Now we had this pixel-based approach. Then we kind of wanted to, we utilized the pixel-based approach to do the computational analysis for ICC networks, but then we again tried to do some refining on that, and we found that we came up this algorithm, this method of like triangulating the ICC networks in order to reduce the total number of solution points, because the overall the resolution requirement is still the resolution is still very low for proper convergence to happen. So what we did was we found the isocontours of the entire image. We triangulate the image using open source available software called Triangle. Then we apply a Delaunay triangulation. Again, basically two-stage. It's not required two-stage triangulation, but we do a two-stage triangulation to kind of make the elements, individual triangular elements, more robust and such that the solution accuracy can be improved. Okay, so there was supposed to be 2 videos running in here. Good. Okay, so that was actually comparison of the pixel-based activity and the unstructured triangular mesh-based activity. Okay, so we actually calculated the entire plan over here is to compare the differences between the pixel-based and unstructured network, and we kind of observed like a same. So what we did was we initiated an activity over this region, which was whereby the activity is propagated towards radially to other parts. We took an average of the entire VM for the both pixel-based and the unstructured mesh, and we found that 20 milliseconds difference between the 2 simulations. So we randomly choose one point around the center, and we found that the activity is around same at that point, but the reason for this difference is because of the conduction velocity that happens, the conduction velocity difference between the 2 simulations. So as you can see, this is the activation time map. So as you can see, like it ranges from around 650 to 600, so around 50 millisecond difference, maximum difference is seen for the activity to propagate from this region to that region. But again, there is, if you ignore that difference, which is actually, if you consider 20 milliseconds difference, that is over the entire average network onto the total activation time of around 500 milliseconds, that's the time when the entire network is activated, that's considered very small. So if we can ignore that, then we could consider something very interesting, and that is the efficiency. So the simulation time for when we consider the pixel time is nearly, so it took like about, so it was simulated on the NESI pan cluster, so about 110 minutes for the pixel-based simulation on 48 cores, and for triangulation method it took nearly 16 minutes, so nearly an efficiency of 5 times, yeah, around that, 8 times actually, yeah, 5 times, okay. So in brief, a framework for modeling the slow wave propagation over ICC network was developed. We observed that a pruning-like mechanism as seen in central nervous system and is hypothesized. We also developed an unstructured mesh-based method, more 80% more efficient, but we haven't utilized that anywhere, and nor did we go back to the postnatal development network anyway. So again, there was a difference in the activation time, but that was only the 4% of the total activation time. So moving on to the three-dimensional aspect. So we have the ICCs, but then for interacting with the smooth muscle cells, as we have seen in around the second slide or something, we see that there is a gap junction between the ICC and smooth muscle cell. So in order to define a continuum equations to define that particular current exchange, we had to define a new model. So we call the multi-cell tri-domain formulation, which is based on a previous model that was developed by the group at Singapore. So in this case, we have two intracellular space and one extracellular space for when compared with the bi-domain formulation. So in this part, in this region, in this the coming slides over here, we develop this model. We formulate that equations. Then we develop an anatomical mesh for simulating the electrical activity, whereby we, in the context of intermingled ICC and smooth muscle cells, we also include anisotropic propagation and model the effect of external pacing onto the normal activity. So as I said, so there will be one extracellular space which will be shared by two intracellular. So ICC, smooth muscle cell, and this will be the gap junction based current that will be flowing between these two. So the tri-domain equation that we developed, that we derived, was symmetrical, positive, semi-definite. So it enabled us to choose a robust solvers for solving such equations. So previously, when they had the extended bi-domain formulation, we had the problem of the metrics not being symmetric, and we were limited with the choice of GMRS solver, which is considered to be inefficient. So that's one thing. So this is the comparison between the two models, the previous model and the current model. So it's the solver solution time. So again, the two different solvers were used in this case. So the top one is the previous formulation, and we didn't have the choice, so we used the GMRS solver. And the bottom, you can see the conjugate gradient solver with the Bloch-Jacobi preconditioner. So you can see that nearly it's around 4 times efficient. So next one is the generation of the entire stomach geometry and the mesh. So as we usually do, we have the CT images. We obtain the surface image using the CM-GUI tool. From there, we export the surface information as an STL. We perform a convergence analysis using one-dimensional network propagation, and based on the convergence analysis, we generate a tetrahedral mesh using the tool TEDGen. So it's freely available tool. So the convergence analysis, we've observed that mesh resolution of at least 0.4 millimeters is required for accurate solution without messing up the solution process. Now, once we have the mesh, the next approach is to get the fiber informations, right? So we need to define the principal conduction axis for the propagation to happen, which actually acts onto the conductivity tensors that we give as the input. So what we did was we actually solve a Laplace equation. So we put boundary condition onto two ends of the stomach's geometry, one high and one low, and we solve a Laplace equation at the generalized Laplace equation over the entire volume. And similarly, for the cross surface gradient, we actually apply the low and high inside the this surface. So this is inside, and this is the outer shell. So once we solve that, and we found the gradient of the entire solution, we get the fiber for as required. So this is longitudinal, and this is the circular fibers for the slow wave simulation. And then this is the simulation that we perform. So we initiate pacing over here. Normal activity starts at this region. This is the fundus. It forms a ring and then it propagates slowly downwards. But then this pacing activity is, as you can see, it kind of takes over the incoming electrical activity from the corpus region. So this is 150 seconds of simulation. So there were 2.5 million tetrahedral elements in the entire stomach mesh. So this was one of the unique model in a sense that it was capable of simulating anisotropy and also gastric pacing simulation. So it took approximately 7 hours to solve around in 128 cores. So we used the NESI pan cluster. So we used the OD solver as a forward Euler and linear solver, block Jacobi precondition, conjugate gradient solver, so which was found to be the best one. So this is the for different pacing activity, how much you can entrain the normal slow wave activity. So that's the analysis that we done, and we found that that it is the pacing protocol is limited by the native frequency as well as the refractory period of the underlying cell activity. So these two factors limit the number of pacing that you can apply for the simulation for the pacing. So over here we develop a 3D stomach model with anisotropic propagation for studying gastric pacing. A novel tri-domain formulation is presented, which is numerically stable and efficient, and which, when applied with extracellular perturbations, the pacing protocol, we found that the pacing protocol was limited by the frequency of the native activity and also the refractory period of the underlying cellular activity. Future works. Now for the cell model that we developed, it actually during the passive state it only considers the cell, the membrane capacitance when one cell interacts with the other one. So we want to include more information onto that. So that's another field, another direction that's from over here. But the literature studies onto that is currently very limited. Then the functional impacts of the ICC network degradation could be investigated using larger networks. We have to actually validate the ICC networks with calcium imaging data, but we still have the issues of the temporal and the spatial scale that we consider for our study. Then the ICC network simulations that we performed can be generalized onto 3D networks. So we do have like 3D images which can be stacked together, and we can perform instead of iso contours, we can get the iso surfaces, and we can generate the tetrahedral mesh onto that. And then finally onto the whole organ level, we could so in this case all the material properties and everything was spatially constant to make things simpler. So we aim to induce dysrhythmia and include spatially varying, like include different muscle fibers for the simulations. I would like to thank my supervisors. They are the best supervisors, I should say, Leo and Mark. Then the gastrointestinal system group research members, especially Dr. Greg, Dr. Peng, Dr. Nira, Dr. Tim, and others as well. Then Dr. Joe, Dr. Gary, Dr. David from University of Oxford for their help with the solvers and chased computational library. Dr. Alberto and Dr. Martin Buist for their help with the developing the tri-domain formulation. Professor Jean-Marie for providing us with the ICC network imaging data. Funding, Ridet Institute, New Zealand Health Research Council, NIH, and the Auckland Medical Research Foundation. Travel Award, Maurice and Phyllis Baker Trust for providing me. They were very generous with me. They provided funding for 2 years actually, 2013 and 2015 for attending conferences, and FEPS Travel Award in 2014. Thank you all for your attention and question and answer. Yeah, that's a distraction so that you don't ask me questions.
So questions for Shameer? Ian?
So how is the whole thing wired? We've got this, when you showed the opening movie,
Yep.
there was this circular wave moving along through the stomach. So basically everything's firing together in one level and then moving forward slowly by this regulation pattern that you're talking about here, right?
Yes.
So that's all to do with the wiring of the nerves around the stomach, is that right? You have a fast conduction pattern circumferentially,
Yes.
and you've got this almost 1D wave moving along longitudinally, is that right?
Yes, that's right. But then you have like, that's a very good question. So you are asking like if it is the same throughout the entire duration. Is that the question?
Well, it just, I mean, circumferentially the wave goes down the stomach,
Yes.
or up the stomach.
Yeah.
And so you've got this really, like everything's wired together at this level here. But then this pattern generator that you're describing basically regulates how fast the thing goes down, which is quite slow, longitudinally.
Yes.
Contraction.
Yeah.
Right?
Yeah.
Okay, so I'm just trying to get my head around what's actually happening.
So okay, so just give a brief thing. Like again, so there is a. I'll just go back to the end. Maybe where is it? Where is it? I trust my simulations. Okay, so this is the fundus region where we store the food that we eat. This is the pacemaker region inside the stomach. So there will be a group of cells which will be actually having a higher intrinsic frequency. So that will generate an activity which will be spread across circumferentially, as you have said. Yes. Then that will be propagated, and that will be controlled. Controlled propagation is seen downwards. You have the ICCs not just over here, but all over, especially all over the stomach. So which had to coordinate the entire activity. So that's where, you know, like if we have ICCs damaged over this region, for example, say this ICCs get damaged. So these ICCs along this region may activate based on its intrinsic frequency and help the propagation to continue rather than, you know, making the stomach die or something like that. So it's like everything is coordinated. We do also have the dysrhythmic activity, so which can like kind of look quite weird, and we wanted to understand that as well. So yeah.
So I guess he was asking, is that pacemaker area a point, not the whole ring around?
So it's not like a pacemaker, it's not like a node concept that we have. It's like a collection of nodes maybe, but they might activate, or if that doesn't activate, maybe neighboring cell may be activating over here.
But it's a zone like in the cluster, not a whole ring.
No, not the whole ring, not the whole ring. No. This is like this simulation is like very idealized in a sense, you know. Yep.
So do we have different kind of ICCs that may affect the pattern of activation? So we have like or we have different density of the cells among the stomach, or is it when you record the activation you'll find it quite symmetric propagation of the action potential? How really it is? How many cell types do we have?
That's a very good question. So we do have many types of ICC. So there are like ICC myoenteric layer, one name, ICC deep multiplex. So that will be inside the muscle layers. Then we have ICC intramuscular. So all these three different ICCs will have its own functions, which is quite critical for the proper functioning of the entire stomach as as a team, you could say that. Now, for the propagation, ICC myoenteric plexus is the one that plays a key role for the propagation, like from the for generating the activity and to propagate along the stomach. ICC myoenteric plexus is the one that actually plays a key role in there. Yeah.
Another question, yeah.
When you had the structured and unstructured triangular meshes, did they start from the same size of edge around those boundaries? Like are they just as dense at the fine points? Yeah, so if you go back to the actual mesh of that, is the element size at its finest on the right-hand side the same size, at the bottom, the same size as at the top?
These ones. This represents the same dimensional traction.
I'm just saying the smallest elements in the bottom and the elements in the top, are they the same size, more or less?
I don't think so. It's a bit bigger as long as it conforms the network contour.
I mean, I just feel that it would be nicer if you had some kind of convergence study on that, because you're talking about however many milliseconds difference. It's almost meaningless without knowing what the right answer is.
Yeah, exactly. That's a very good question. We still don't understand what's the difference between these two. But to say about the convergence analysis, what we did was we further refined this network to a very fine resolution, and we still had the unstructured network. When that case happened, that I would at that stage, like when it was very fine, the size was almost similar, but then again we saw like 20 milliseconds difference. So I think it's rather related to the regularized nature of the
top one as well?
Sorry?
Did you refine the top one as well?
Well, it's already very, very small. it's actually like well, if you would suggest that probably this is not converged. Do you reckon that's no? Okay. So, well, we try to reduce the size of unstructured mesh onto like a very small size, but then again there was a considerable difference. Yeah, but we didn't reduce the size of this one, but as we assumed that might be converged. So yeah, probably that should be the next step we should do actually. Yeah, thanks for the session.
What's the model of the whole stomach with the so the images didn't have fibers in them, and you generated the fiber directions from the geometry of the stomach.
Yes.
How accurate is that, and how sensitive would it be? Because I remember that you had a slide where you had a blue and a red bar on the top and bottom of the stomach, and the blue and the red bar on the inside and outside of the stomach.
Yes. Oh, okay. So the stomach, if you consider that's a volume, right? And you have a surface at the, so I can't basically show that over here. So there is a small ring of surface over here at the very edge, and over here as well there is a small ring of surface. So if you consider as a 3D, then you have like 8 different surfaces, right? No, 6 different surfaces.
So those surfaces were visible on your surface reconstruction after the...
Yes, exactly. So we prescribe the boundary condition only onto these surfaces, and also again for the inner surfaces as well. Like so, this is for the entire mesh geometry as well. Yeah.
Jenny?
What's the ratio of your electrical anisotropy like between the circumference and longitude?
That's a very good question. It's quite high. So it's like around 20 or something.
Right. Do you expect to see like a correlation of that in your histological images? Like for example, more connections in the scapular direction than there is in longitudinal?
Okay, so that's a very good question. So we believe that that's because of this anisotropy is because of the muscle layers that have to play a role into it, because the muscle layers are embedded with the ICC deep muscle layer plexus or something like that. Yeah, I'm sorry, DMP or something like that. And so that kind of like helps into the spread of the slow wave activity onto the entire circular region. So I would assume that's because of, but that's a very good question, and we have to actually that's kind of like, so we haven't quite delved into like importance of muscles onto the slow wave propagation, but yes, that's probably the next one.
Any final questions? All right, so let's thank Shameer